Hello and welcome from Houston. My name is Christoph Eichen. I'm the head of technical services here at LC Sciences and responsible for the microarrays division. If you were earlier on one of the webinars today, we had one for Asia Pacific for that area. There I was joined by Dr. Julie Lang. She's a technical director for our China office called LC Bio. Just a few notes. We will make a recording of this webinar available, as well as a PDF file format slides. And the PDF file will also include all the links, so you don't have to write down any links that I'm mentioning here. During the webinar, of course, you feel free to enter any questions into the chat box, and Jason and Chris, who are here with me, will be trying to answer them as we're going along. And at the end, of course, there will be time for any additional questions and answers. Okay, just briefly agenda. I will, as outlined already when you signed up for this webinar, talk briefly about microRNAs, specifically what we know about microRNAs in plants. Then highlight or briefly mention 3 recent studies or publications that came out early this year, or one of them actually came out last year. And then talk about current methods, mostly for discovery and profiling of microRNAs, and then end with a couple of case studies, which are basically customer publications. And while we won't have enough time, of course, to go into detail of these case studies, I hope it will give you an idea of what is being done in the field, and hope will serve as inspiration. If you're new to the microRNA field, chances are you're not familiar with our company. Just briefly, our headquarters is in Houston, Texas. We have also a rather large office in China. And we have representations in Japan, Korea, and India. All the lab work is being done here in Houston, so samples worldwide are ending up here. We were actually the first microarray service provider, so we're doing this since 2005 and have processed thousands of samples. The company was founded around our core technology, which is a unique microfluidic chip technology. There are a couple of things that are unique to it, but the bottom line is that it allows us to easily customize any type of chip. So basically all chips that we synthesize are custom synthesized to order once we receive the samples. To give you an idea about who our customers are, we have customers in 40 different countries, and typical customer is academic, but we have an increasing number of biotechnology companies in the pharmaceutical field, as well, of course, as research hospitals and government agencies like the Food and Drug Administration or the US Department for Agriculture. As mentioned, the company was founded around the core technology, the so-called microfluidic chip technology. And it's basically chip technology that allows us to synthesize any type of RNA, DNA, or even peptide on the chip surface. And in 2004, 2005, the founders were looking for a good application for this technology. And fortunately, they were visionary enough to realize the potential that microRNA showed back then already. And so microRNA, microarrays were basically the first application. And we've been, since that time, we've been so busy with this field that we've kind of neglected the proteomics approach or proteomics applications that we can use this technology for. But this is something that we're trying to focus on in the future. Okay, microRNAs, what do we know about them? First of all microRNAs are small, non-coding RNAs, typically consist of 20 to 22 nucleotides. That would be the case for animals. For plants, they're typically a little bit longer, up to 24 nucleotides. All microRNA precursors have a well-predicted or well-defined stem loop region, as well as a hairpin structure. This secondary structure is actually very, very stable. It's under the typical hype conditions of a microarray, it doesn't unfold. So you will not be able to detect any precursors on an array typically. Another thing is that many microRNAs are evolutionary conserved between species, and the species can be as distant as, let's say, worm to human. This is also, of course, the case for plants. Number 4 is basically that it's known that microRNAs bind complementary to messenger RNA and act as negative regulators of translation. And there are basically two different mechanisms depending on whether it's in an animal model or in the plant. And I will mention that we'll go into detail there in a little bit. Fifth is that they exist in a very high copy number. And 6 is that their microRNA expression is tissue, developmental stage, and also, of course, disease specific. This is the reason why we can use microRNAs for as biomarkers, as disease-specific biomarkers. Currently, as of today, there are 21,643 mature microRNAs from 168 plant, animal, and virus species in the publicly available database called MIRBase. This database version right now is version 18. It was updated in November of 2011. From what I've known is that there will be an update to version 19 any day now. I was told that it will be either end of June, early, or first half of July. The mechanism of microRNAs is rather complex. This is because each microRNA can control many genes, and also the other way around. Each gene can be controlled by multiple microRNAs. So it's not a simple one-to-one equation here. For the human genes and the human body, it's estimated that up to 1/3 of all human genes are regulated by microRNAs. As mentioned earlier, they might operate based on two hypothesized mechanisms. One would be, which is predominantly seen in plants, it's the so-called complete pairing. Basically, the microRNA binds to the messenger RNA, and you have a perfect match. And the result is that the messenger RNA gets degraded. In the animal model, you have you don't observe this complete pairing, but instead you see so-called imperfect pairing. So the microRNA binds to the messenger RNA, and it's not a perfect match. There are some mismatches, and the binding is strong enough to repress translation, but not strong enough to trigger degradation of the complex. Already in 2009, a paper in Cell mentioned that based on the sheer abundance and diversity of the plant microRNAs, and specifically probably each and every single gene in plants is regulated by microRNAs during the developmental process at one point or another. To give you an idea about the time frame that we're talking about here and how fast this field is developing, 1993, Lin4 was the first microRNA that was found, and it was actually found by Victor Ambros, Rosalind Lee, and Rhonda Feinbaum. And it was found in C. elegans, and it was not really clear. It was considered not a fluke, but a really a rare occurrence, and nobody thought about that there will be that we will have maybe 2,000 or more human microRNAs one day that we will find. And it was, of course, a major challenge to the central dogma of biology, which basically is founded on the principle that DNA gets transcribed into RNA, and then RNA gets translated in protein. And in that dogma, there was really no space for a place for a small RNA molecule that actually regulates that transcription. So 7 years later, in 2000, LED7 was identified by the SLAC lab, and it was found in human as well as in Drosophila. And from then on, basically, things moved on pretty quickly. 2001, Bartel, Tuschl, and Ambrose, these three groups discovered a large class of small regulatory RNAs and named them microRNA, hence the name. And the same year already, the Bartel lab discovered microRNAs in plant, and they described 16 different microRNA sequences from Arabidopsis, and by doing a sequence comparison, they were able to show that 8 of these 16 microRNAs were conserved in the rice genome. Then 2002, already the first, basically the microRNA biogenesis pathway in plants, in this case for Arabidopsis, was published. Again, then the Bartel, Zamora, and Reinhardt in 2003 already described the microRNA silencing mechanism for plants. Outside the plant field, things developed equally fast. So in 2004, it was already linked to the neuroscientific field. In 2005 already, the next-gen sequencing was used for the first time for the discovery of small RNA and the analysis. So back then they used Solexa, which is nowadays Luna, sequencing to identify new small RNAs in Arabidopsis. Then 2005, this is exactly the time point when LC Sciences was founded. The three big studies linked microRNAs to cancer. 2006, the cardiovascular field followed. In 2006, the MIRBase, the publicly available database, went online at the Sanger Institute. It was founded earlier in 2003, 2004, but this is when they went online. And this is today still the main database that everybody looks at, and that is functioned as a depository for experimentally verified microRNAs. In 2008, the plant microRNA annotation was standardized. In general, the microRNA nomenclature is kind of fluid, let's say. The latest update was done with the MIRBase update in November of last year to version 18. I'll mention some changes in the following slides there. Another important database is the so-called plant microRNA database, which went online in 2010. It's hosted by the China Agricultural University. And what this includes are not just the experimentally verified microRNAs, but also data that are generated in-house as well as predicted sequences. Briefly about the processing of microRNAs. So after the initial transcription from the gene, they exist as the so-called prime microRNA, which is basically the primary transcript. This transcript has already, of course, the stem loop structure and the hairpin structure. Indicated here is also mismatched regions. It also has a 5' cap modification as well as a poly A tail. This primary microRNA transcript gets processed by protein complexes called DORSHA and PASHA. What happens here is that basically the 5' cap gets cleaved off as well as the poly A tail. The resulting molecule is called pre-microRNA or precursor microRNA. All this happens in the nucleus, and this precursor molecule then gets transported into the cytosol. Once in the cytosol, it gets further processed by a dicer molecule, and basically what happens is that the hairpin structure gets cleaved off, as well as some modification is being performed at the 3' and 5' end. The resulting molecule is basically you're left with the stem loop structure, basically the microRNA star duplex. The microRNA star is the antisense of the microRNA, of the actual microRNA mature form, and it's actually was just discontinued with the latest nomenclature change. So microRNA star was typically the one that was less abundant, or was that form that was found second. So the first form of this duplex that they found that was sequenced or verified was typically called, let's say, microRNA number XYZ. And the complementary strand, the antisense strand, was then called microRNA XYZ star. Because it was always unclear which one was which form from 3' to 5' or the other way around. The nomenclature was recently changed, and so the star nomenclature was retired, and now each microRNA mature microRNA is called either 3P or -5P. These changes were implemented already for human, mouse, and C. elegans for the other species that this will follow. Initially, the idea was that only one of these two molecules that are present in the duplex are actually functional. It turned out to be that both actually can be functional. So for the purpose of the slide here, we will go just ahead with one mature form. Typically the microRNA without the SAR, basically the sense version is called the mature microRNA, which then forms the microRNA-induced silencing complex, or called myRISC. The antisense part of the mature microRNA is typically degraded quickly. As mentioned, there are cases where this form also can be functional. For the latest updates regarding the microRNA nomenclature, I can recommend to look at the Mairbase page. There's a blog section where Sam, who's maintaining Mairbase, has written some very good articles about the nomenclature and what the name of the microRNA actually includes. Briefly, the difference between the animal model and the plant system is shown here. It's on the left side as A is the animal model, exactly how I just described it. And on the right side here, under B, in the right column, you see the process in the plant system. The main difference is that all the processing is done in the nucleus and not in the cytoplasma. So the processing and the editing is from primary transcript to precursor microRNA to the microRNA star duplex is performed all at once in different steps, of course, but it's all done in the nucleus. This is basically the main difference. Besides the processing, there are some other unique features about plant microRNAs. First of all, they are basically near to perfect complementary to the messenger RNA. This has the advantage that when you're trying to predict or identify microRNA targets in plants, it's relatively straightforward as compared to the mammalian system, where you have mismatches allowed. Then, as mentioned before, in plants, the microRNAs since they have perfect or near perfect complementary targets, the binding is perfect and actually triggers complete degradation of the messenger RNA. Number 3 is that microRNAs in plants show more variation than for the mammalian system. This is, for example, also true for when it comes to isoforms of the microRNAs, as revealed recently by next-gen sequencing. Okay, which functions do microRNAs have in plants? In principle, they regulate anything related to plant development. This includes leaf development, floral development, root development, etc. Then microRNAs are also involved in signal transduction. They're involved in response to disease and resistance. A big area of research is the microRNA involvement in the regulation to or response to environmental stress, which can include biotic as well as abiotic stresses. And of course, microRNAs can regulate the microRNA and siRNA biogenesis and function. This graph illustrates the rapid advancement in knowledge about and the number of plant microRNAs. What is shown here on the x-axis are the mir-based versions, so the general publicly available database versions. It doesn't start at version 0 here, but it starts at version 6. But you get the idea here. So from version 6, with 9 species and just a few microRNAs that were characterized, we are now at 51 species and close to 4,000 different plant microRNAs. And all of this in over a period of roughly 6 years. As mentioned before, the other important database is the plant microRNA database. It contains actually way more species and entries. So instead of the 51 species here of version 18 in mir-base, we have here 127 species. And instead of the 4,000 entries, we have more than 10,000 entries. This database gets their information, as mentioned, from some in-house studies, from other published papers, and also from predictions, and combines that with the content of mir-base. And there are a lot of different databases out there, and unfortunately, lots of them get released and then not really maintained. This one here seems to be the exception. It was just updated a few days ago on June 11th, and the update includes the latest sequences from mir-base version 18. As mentioned, this includes also predicted sequences, and the majority of the sequences here in this database are actually predicted. So keep that in mind when working with data from this plant database. Here on this slide, we want to summarize some computational tools that are available for plant microRNA data analysis. And while we started doing this, we realized there's such a vast list of programs and websites out there that we will include here a link in the PDF file so that you can download the entire page. Looking at the recent developments, we looked at 3 different papers here. One is a review about the function of different microRNAs, and especially when it comes to the response to stress, abiotic or biotic stress that the plant undergoes. This paper here by Shankar et al. Is actually an updated version of the earlier review paper from 2007. And just to give you a brief idea, it basically summarizes what is known currently in the field. And on this slide, for example, it shows you in this table, in the very left column, it lists all the different microRNAs that are known to be differentially regulated. And then here across the top, it lists the different types of stress that this differential regulation is observed. In each of the boxes here, then it shows the different species. Let's say, of course, Arabidopsis here, it's upregulated, and how the up and down regulation is across different species and different types of tissue. So if you're starting out and interested in stress response and how microRNA is involved in its regulation, this is the perfect starting point. Then a paper that was published last year, actually, in I think it was September 2011, at least the online version showed up then, really accomplished something that not all papers get accomplished, which is that microRNA really hit the mainstream media as a result. Basically, what this paper showed is the authors showed that microRNAs from plants can regulate functions in the human body. In that particular case, what they looked at are patients that were eating rice, and they were able to find the rice microRNAs in the human body, and which genes it might target. This would be the first time that cross-kingdom regulation is observed, and a lot of people are very skeptical of this, and so far it has not been reproduced. It has been known since a longer time, 2 or 3 years, that microRNAs actually are being transferred via the breast milk from the mother to the newborn, and can regulate the functions in the newborn, which makes perfect sense, of course, when you think about how important breast milk is to the newborn. People were skeptical there initially too, because how in the world can microRNA be so stable and can be transported from even within the species, and then in this case of this paper, even survive the digestive process in the human body and end up being so stable that it still can regulate genes. So there's a lot of skepticism around this paper, and it hit the mainstream media because a food critic actually saw this paper and didn't quite understand it, and published something about microRNAs and genetically modified crop or genetically modified food, how genetically modified food includes microRNA, and that basically microRNA are bad. That was the really short version of the paper, and it got picked up by a lot of news agencies, and the scientific community reacted pretty promptly, and within less than 24 hours, I think the author had to rewrite the entire article. And if you want to follow the discussion, I put the link down here, as mentioned in the PDF file. You can just click on it. This is a blog from a biologist who tried to summarize the paper and set the record straight, and basically just focus on the facts. And yeah, while it's certainly interesting to look at the effects that microRNA from the food that we uptake has on our body, that's certainly nothing that's exclusive to genetically modified food. Another interesting aspect is, when it comes to plants, is the use of artificial microRNA to improve the resistance, specifically to viruses. Here on the right in the box, there are 5 papers mentioned that all deal with this. What is shown is that expression of modified microRNA precursors can result in the production of so-called artificial microRNAs, or a microRNA, that's the short form. And these artificial microRNAs then can go ahead and target viral RNA sequences and hence improve the plant virus resistance. Looking at the research tools that are available today, of course, that's not just plant specific, but applies to all fields. Looking at the different categories here, first the identification, the different research steps, basically. Looking at the microRNA identification, you have, of course, genetic screening, direct cloning, sequencing. Then computational strategies, relatively easy in plants versus mammalians. Different software out there, microRNA, Find microRNA, microScan, and so on. Then, of course, what we've done also a lot, tiling microarrays. Once you have the predicted sequences, try to validate them by tiling the sequences on a custom array and see where the binding maximum is. And of course, nowadays, next-gen sequencing. And so for the identification or the discovery step, I will talk about the next-gen sequencing method later on in more detail. Then when it comes to detection and profiling, of course, northern blot is still relatively common. Then in situ hybridization, real-time PCR, of course, that's probably most people are exposed to that. Then, of course, microarray analysis, and some people use also next-gen sequencing for the profiling. Then moving on to the target determination step, it's, of course, typically lots of bioinformatics, then gene and proteome expression analysis, pull-down assays we see, then 5' arrays analysis, and something that we just started offering is degradome sequencing. Then when it comes to the pathway analysis, of course, bioinformatics is essential. We have a site called mirfocus.org where you can enter the differentially regulated microRNAs that you find and then see which potential target genes there are. From what I recall, at the moment, we're limited to only the human species that are there. I will have to double check that. Then when it comes to functional analysis, you can do, of course, luciferase assays, gene knockout or overexpression models, and or you can, of course, use antagomeres and inhibit the microRNA expression. Another point would be then microRNA mimicry, where you're basically mimicking the actual microRNA. So from all these tools, looking at all these tools, I will focus on the following on next-gen sequencing as well as the microarray analysis. So basically, there are typically 3 steps involved, of course, in this field. First of all is the discovery. You want to find the microRNAs specifically, especially because in this field there's still lots of work to be done. There are a few well-characterized plant species, but once you step out of that box, then you're pretty much on your own. Then, of course, once you know your sequences, you can do the profiling, and then later you can quantitate that or validate your results with qPCR. So the 2 better and brother methods, of course, microarrays as well as validation then with qPCR. And that was the typical workflow until next-gen sequencing became available and more affordable. So for the discovery process now, here in-house, the typical workflow is we do the discovery with next-gen sequencing and use then custom arrays that are based on on the next-gen sequencing results to do the profiling, fairly low cost and high throughput, and validation, of course, then still with qPCR. Both methods, next-gen sequencing and microarrays, have their strengths. So we decided to take advantage of the positive aspects of both and combine that to a workflow that we call seqarray. No matter which method you're looking at, first thing is, of course, that you need to prepare your sample. There's a whole variety of kits and methods out there. Of course, there's the good old trisol method. The observation that we made over the years is that people that are familiar with the method can obtain excellent results. People that are new to the field and have not used this method before typically struggle for a long time to get usable samples. Also, you want to make sure that the trisol protocol, if you want to go ahead with that route, that that the protocol is modified in the way that it actually retains the small RNAs. The typical total RNA protocols with trisol were developed before microRNAs were even known, so they typically don't retain the microRNAs. We have on our website, if you go under the frequently asked questions, we mention what you should modify or how you should modify your trisol protocol. But if you're open, we would strongly suggest to pick any of the commercially available kits. There are three main companies out there: Norgem Biotech, a Canadian company. They have a lot of specialized total RNA extraction kits that all retain the microRNA. They have specialized kits for a lot of different applications. Then, of course, QIAGEN. There are two different kits. The one that we recommend is the miRNA kit. The one that we do not recommend is the one that sounds very similar, which is the RNA kit. That one, again, is a kit that was developed before microRNAs were really understood, and so it doesn't retain the microRNAs. So do not use the RNA kit, use the miRNA kit. Then when it comes to Ambion, we have very good results with the microRNA kit. No matter which kit you end up choosing for your experiment, make sure that you use the same kit or the same extraction method for all the samples of the experiment. Because while all work, they all will give you a slightly different profile. So stick to one method, and that way you avoid any additional variation in your samples. As QC step, here I list what we use here in-house for this slide is actually used for mammalian samples, but the UV-Vis ratio is of course also true for plant samples. So you want to check your you might want to check your UV-Vis ratios with a nanodrop or something like that. The 260 to 230 ratio should be above 1. The 260 to 280 ratio should be above 1.8. What is shown here is a bioanalyzer run. We do not run it by default. In our experience, of course, it gives you a good estimate for the overall total RNA quality, especially the larger RNA. But still, it doesn't really tell you much about the microRNA quality at all. And we have had cases where the RIN value was around 3, and so all the large RNA was degraded completely. But the microRNA was still perfectly fine. And we had also opposite cases where the RIN value was 10, but the microRNA was either not retained or was completely degraded. So what we do is we check the UV-Vis ratios and then go ahead with the samples on the array. And especially with first-time customers, to get a feel for their samples. And yeah, if it doesn't look right at any point in time, of course, we would contact you. Just to give you a visual impression of how good and bad samples look like, these are just gels, different types of gels run. Always the good one with the microRNA fraction compared to a poor one that's lacking the microRNA fraction. One of the biggest misconceptions that we see is when it comes to experimental design and the different methods is that somehow different rules of statistics apply to the different methods. So what I mean by that is that we see it over and over again that potential customers that are interested in next-gen sequencing have the impression that you don't need more than one sample per biological group in order to do any type of statistics. Of course, that's not true. If you want to have any type of statistics done, you need at least 3 samples, biological repeats per sample group. So if you're looking, let's say, at stress response through salt and versus normal plant, then you would have to have 3 normal samples and 3 stress response samples. And yeah, this is independent from the method, whether you're using next-gen sequencing or arrays or qPCR. The basic rules of statistics, of course, do not change based on the method. Comparing microarrays and sequencing, we summarized here the key advantages of both. Looking at the microarray, it's a very reliable method, pretty robust. It's been around for a long time. It had kind of a bad reputation because of the early data and the slide quality and the array quality, because the spotted arrays, the early generations showed pretty bad signal uniformity. But that has improved dramatically, and with our technology, we have a completely different system anyway. The high throughput, it's fairly high throughput, can be streamlined, of course, automated, and the data analysis is very straightforward. And the simplified data analysis is also reflected in the short turnaround time. So for us, for example, it's typical 2 to 3 weeks, depending on the number of samples and the workload. But we also offer a rush service for 5 that gives you data within 5 business days. And of course, compared to RNA sequencing, it's much lower cost. And for the actual profiling, we still prefer the microarrays over the sequencing. Looking at the sequencing, of course, you get a much broader, comprehensive picture of your transcriptome, because you can analyze not just the microRNA, but also the messenger RNA, other non-coding RNA. But while that's of course true, is when you look at smaller RNA, you will have to run a different type of sequencing as compared to looking at the gene expression, the messenger RNA level. So typically you would look only on the microRNA level. Of course, the biggest advantage is that you don't have to have any prior sequence knowledge, so it's ideal for discovery. It has an increased dynamic range, and you can tune the sensitivity, and you can detect details about the structural variations like isoforms, alternative splicing events, and such. And in contrast to an array, it's microarray. It's a truly digital solution. That means you get absolute copy numbers and not just a relative abundance. Just briefly, this is our default chip. We have different chips available, but this is our standard chip with 4,000 features. It's pretty much a closed system. You have a silica base where you see here, what is shown here on the right are the individual reaction chambers here. It's an electron microscopic picture. And this silica chip is then covered with glass, and we pump the liquid through through the chip. And in each of these reaction wells, we can synthesize a different microRNA probe. And the way the synthesis works is via photosynthesis, so we are extremely flexible, and this allows us to keep up with the permanent changes in Mir base, and that's what we call Mir base synchronicity. So right now we are at version 18, as mentioned, and the what is marked here by the two dotted lines is major changes in the Mir base versions and nomenclature, or in the probe content, in the sequence content. For example, the first one here to the left is the change from version 10 to 10.1. That was the it doesn't look like a major change, but it basically included lots of corrections and name changes. So if you work with older data, that's something you want to keep in mind. It's very difficult to compare anything that is pre version 10.01 with the later data. And then, as mentioned earlier, there was a major change from version 17 to 18 when it comes to the nomenclature. So typical workflow, just briefly here, is when we receive the samples, of course, sample QC, then we prepare the sample for the hype, for the labeling, do the hype, scan the chip, do the data analysis, and send you the data. This just same workflow illustrated. You typically work with a couple of biological repeats, do the arrays, normalize across all chips, and do then the statistical analysis, and you receive the different heat maps at different with different p-value cutoffs. And this just to illustrate the data quality of the chips. What you're seeing here are these single clusters. These are the probe repeats that we have on the chip. So for each microRNA, we have multiple probes, multiple identical probes on the chip. On the smallest chip, we have 4,000 features. So and we always fill the chip up. So if there are only, let's say, 200 known sequences of your species, we fill it up nevertheless, and you get a very high number of repeats, of course. Yeah, this is just illustrating what that you basically we will send you raw data as well as fully analyzed data, so you will be able to go back and reanalyze all the data from scratch if you choose to do so. Looking at the advantages of RNA-seq or next-gen sequencing for microRNA discovery, it's of course the main point is really the discovery. The drawbacks of the next-gen sequencing that we feel is that the sample prep is still very involved, whereas for the microarray, you're using the sample straight as it comes without any amplification steps, nothing like that. And here for the sequencing, it's a rather involved sample prep, and chances are that you're, with all these amplification steps, etc., that you're biasing your profile. So we've heard it numerous times that people have sometimes trouble to actually validate the sequencing results with qPCR versus an array, which has typically a much higher success rate. So when it comes to discovery, we recommend sequencing. When it comes to profiling, we still prefer microarrays at this point in time. Workflow is pretty much the same thing. Sample QC, then sample prep, the library preparation, then the actual sequencing, and of course the bioinformatics, and then and data analysis according to the customer specification. There are, of course, different sequencing platforms out there, which the one that we're using is the Illumina platform. We have using different machines depending on the application type, but for the RNA-seq, for example, we're using the high-seq, of course. Then for the typical microRNA discovery run, we use the illustrated machine here. It's an Illumina genome analyzer, GA2X. The read lengths are 35 bases, and you typically get up to 30 million reads per lane, that is. And you can, of course, barcode or multiplex and run multiple samples in each lane. The number of samples that we would recommend to use per lane depends really on the goal of the experiment. If you want to really discover new microRNA sequences, we would recommend, of course, a lower number of samples versus just a profiling experiment. So typically we recommend up to 3 samples for discovery, and then when it comes to profiling, you can add more. That we would not recommend to go higher than 6 samples per lane. And the table just illustrates, of course, the basics, that the more samples you're adding per lane, the lower the number of reads you're getting per sample. And yeah, for example, when it comes to discovery, you might even want to run only a single sample, but maybe a pooled sample of different developmental stages to get then the highest number of reads and get even low abundant microRNAs. Yeah, this just illustrates that the basic package that we offer is the basic data analysis package that's included by default. Includes Illumina base calling and analysis of using the Illumina package, and also using our own software, doing some filtering and reduce the process data to mappable reads. And when it comes to the advanced bioinformatics, we map basically it's smear-based, the genome, related genomes, and the goal is to get as many mappable reads as possible, of course. Then we do the alignment, classification, the functional annotation of the reads, as well as then also predict novel microRNAs, of course. And if you choose to do differential profiling, we do this as well. This I just included so that you have it in the PDF file. This just outlines the data flow of the bioinformatics step using our own software in a little bit more detail. So what type of information are you getting from next-gen sequencing? Because besides the copy numbers, of course, and the actual sequences, this is another example. It's basically shows you the length distribution of the mappable reads. I mentioned earlier, plants typically up to 24 nucleotides, which is exactly the case here. Then very interesting are the so-called isoforms or isomers. There's always discussion about how to which extent these isoforms, these isomers, are actually real, or to which degree they might be just artifacts of the extended library prep. What is shown here for sure is that you have one predominant form, which is shown here in the first row with almost 60,000 reads. And then you have a second isomer that is truncated by one nucleotide, and it's still pretty abundant. There's still more than 10,000 reads here, and all the other isoforms are rather low abundant. But these isoforms are certainly also a problem when validating data. When you run an array based on probes that are designed around the sequence, the main form or one of the isoforms, a microarray will not distinguish between these, between the truncated version and the full-length version. Whereas, of course, something very specific like a TACMAN microRNA array will distinguish between these two. So that is one of the reasons probably why you sometimes have trouble validating array data with qPCR data. So both, as mentioned, both methods have their strength. Since we feel that for the profiling, for multiple reasons, cost, turnaround time, ease of data analysis, as well of the fact that you're using the sample directly without any elaborate library preparation, which might bias your sample profile. So all because of all these reasons, we prefer the microarrays for the profiling still. But then especially for rare sequences, rare species where only few microRNA sequences are available in MirBase, we use typically a pooled sample of different developmental stages of that species and sequence this, oftentimes as a single sample per lane. Then based on these data, we then create a custom array chip, custom layout, and do the actual profiling then using these custom chips. Now the whole workflow is outlined here again. So here in the blue first column, it illustrates the deep sequencing steps. Then in the second one, it's the bioinformatics work that basically leads to the identification of novel microRNA sequences in your sample, as well as combine it with the known sequences and develop the custom array based on that information. And then here in the third column, the custom microarray workflow where you do the actual profiling using multiple biological repeats. So this way you're taking advantage of the latest technology when it comes to the discovery step, and you take advantage of the cost effective profiling using microarrays. And this method we've applied now multiple times successfully, and these are a couple of the publications in the plant field from last year. So shown are 4 different species here: barrel medic, poplar tree, soybean, and rice. In each of these cases, we identified new microRNAs. Of course, more in the rare, in the less well-characterized species. Soybean was already well-characterized last year, so there we found only 8 new microRNAs. But in the other cases, we have in the first 2, for example, we found more than 50 new microRNAs by sequencing. Combine that this information then with the known microRNAs from the database, and create the custom arrays, and did the profiling as outlined earlier. And in each of these cases, in each of the publications, the customers were able to reveal, in the first case here, 12, in the second case, 27 differentially regulated microRNAs. So I touched already on these topics, why you would want to study microRNAs in plants, but basically there are 3 major topics: basic research, of course, the discovery of the microRNAs and the pathway, then the stress response studies, biotic as well as abiotic, and of course disease-specific biomarkers. This slide is just to illustrate that some of the species that we worked with. Soybean is of course not that exotic, but coconut, rapeseed, tomato, rockcress, rice. Of course, we've done citrus plants, and whole variety of different plants. And this paper here is an example of for typical basic research or discovery study. Basically, this was done for tomatoes, and in this case, it was done with a microarray, which was designed the way that 13 precursor microRNAs that were predicted were tiled on the chip, their sequence, because from the precursor microRNA you still don't know how exactly the mature microRNA looks like. So different potential mature microRNA forms were synthesized on the chip and then hybridized with a tomato sample. And 78 highly expressed mature microRNAs were detected as a result and verified. Then this was an array example, and then here something similar was done with sequencing, was microRNA sequencing for the discovery step. In this case, novel microRNAs from anti-targets in cucumber leaves and roots were discovered. Here in this particular paper that just came out this year, a total of 29 known microRNAs and 2 novel microRNA families with a total of 64 new microRNAs were identified. And then the sequencing data were then followed up with qPCR analysis to look at the differential profiling. In addition to the actual discovery step, this paper also includes the target analysis using genome sequencing. So basically, this paper includes all 3 steps: the discovery, the validation with qPCR, as well as then the target identification using genome sequencing. Moving on to the second topic, it was the stress response studies. In terms of biotic stress, we have seen samples regarding viral infection. I'll mention a paper there. Then to fungal infections and bacterial infections. These are the 3 main biotic stresses. Then regarding abiotic stress, we looked at salt, or our customers, I should say, looked at salt, temperature, then water, whether the plant is submerged or not, drought. We looked at nutrient deprivation, phosphate, sulfate, copper, and nitrogen deficiencies were looked at. And of course, other stresses as well, chemical exposure or pollution in general. A nice paper about the stress response study, which involved arrays, is shown here. This was published in 2011. And it looked at the tomato response or the regulation by microRNA once the tomato plant is infected with the cucumber mosaic virus. And you can see here very nicely, this is the array, this is the control array, this is the infected array, and you see how certain microRNAs are completely missing. These 4 microRNAs in the very right lane are missing in the second sample here. And the shape here might look unusual to you. These are basically the raw images of our chip. So we have as mentioned, we have a microfluidic system, so we don't have the typical round-shaped spots of spotted arrays. We have a reaction chamber that has this elongated shape. This is why you see this shape. And then here the table is pretty much looks like it's not really legible here, of course, but it looks like the table that we're actually sending out in the data report in terms of what is up and what is down regulated. And here again in the heat map. Here are the heat maps that illustrate the same effect. Here's another stress response study regarding abiotic stress, in this case, salt tolerance in maize roots. Here, a total of 98 microRNAs showed an altered expression. This was also done with a microarray again. And not only that, but the authors then go ahead and hypothesize about how these microRNAs, what they target, which genes they target, and how they actually might basically in a concerted effort, regulate the response to the salt stress. Here's something similar done in not with an array, but it's a stress response study looking at the response using sequencing. Here, they looked at the long-term effects of waterlogging of maize roots, and a total of 61 mature microRNAs were found. Some of them were known, but some of them were newly discovered. So total of 25 potential new microRNAs were actually discovered, and a total of 32 microRNAs were shown to be involved in the response to waterlogging. So those were shown to be differentially regulated. And the targets of these microRNAs were then, in the second step, identified using degradome sequencing. Another study was done with sequencing. Here, the authors looked, also a recent paper of this published this year in PLOS One. The authors looked at the nitrogen deficiency in maize roots, and they discovered a total of 99 new loci, which belong to a total of 47 new microRNA families. And here in the two graphics here to the left and to the right, it's again the microRNAs are listed here to the left, and then the fold change of these in terms of up and down regulation is illustrated. And as last example, it's a plant breeding application example. It's basically where the customers looked at different plant mutants and looked at the effect that the mutation had on the microRNA expression levels. And of course, that is a big field in trying to get plants more drought resistant or more resistant to viruses also, of course, like mentioned in the other examples. So we are located in the United States. As I mentioned, we have offices in China and representations in India, Korea, and Japan. And so all the samples are being processed here in-house. And yeah, so I hope this was helpful to you and somewhat interesting and gave you maybe some ideas. If you have any questions, please feel free to ask them either now in the chat box, and we will get back to you. We will stay on for another 10-15 minutes here to make sure that we get to that. If the questions are too specific or we have to touch base with the lab, then we will email you, of course, personally. Other than that, if you have any other questions or if you're listening to the recording here, please feel free to email us any questions that you have at service@lcsciences.com. Thank you again for your time. I hope it was helpful, and have a great day. Thank you.